Hello, everyone, and welcome back to the fourth session of our workshop. I'm Tachi Yamada, and along with Ann, our co-leads for this particular initiative. As you recall, the first three sessions have dealt with basic science, technology, and implementation science as related to enhancing healthy longevity. We've had a very robust and extensive discussion on all three topics. But having completed all this, it seems to me that it would be useful to look at all of these areas in somewhat of an integrated way, and also to make sure that we've covered all the bases. So this last session is really to look at gaps in our discussion, things that we haven't thought about, things that we need to think about, and understanding what the opportunities are that we haven't covered as of yet. We have three speakers to cover each of these sections to set up the discussion. Our plan is to have each of the speakers speak at the front of the session and then have a broader discussion on all these areas following the speakers. The first speaker, who will speak on the basic sciences or biological sciences, is Dr. Tom Chung, who's currently the SHO Associate Professor of Life Sciences in the Division of Life Sciences at Hong Kong University of Science and Technology. He's the director of the HKUST BGI Joint Research Center and the HKUST Nanfang Life Sciences Joint Laboratory. He's also a member of the Hong Kong Center for Neurodegenerative Diseases, Center for Stem Cell Research, the Center for Aging Sciences, and the State Key Laboratory of Molecular Neuroscience at HKUST. Professor Chung is a skilled and accomplished stem cell scientist and researcher on the biology of aging. The main focus of his laboratory is to specify the molecular pathways that control stem cell quiescence and stem cell mediated tissue regeneration to achieve a better understanding of somatic stem cell function in the context of tissue regeneration, diseases, and aging. Chung Lab has solid experience on mouse genetics, next generation sequencing, single sequencing, and bioinformatics platforms. So without further ado, Tom?
Can you hear me okay?
Yes, we hear you fine.
Perfect. So let me share my slides. Okay, I suppose you can see that full screen. Well, I'll say, first of all, this is Tom. Thank you, Anne and Tachi, for the opportunity to have the chance to participate in this wonderful workshop and to have a chance to speak here. So in the first section, Eric has started with a great summary, and we also had a wonderful discussion with the panel experts. And I think we cover up good aspects of various different biological sciences regarding aging. And for me, as a biologist focused on aging research, we understand that the aging experiments require very careful planning. And in my short presentation today, I would like to kind of set forth a discussion point regarding some of the considerations in data collection, and which I think it is something very important for aging research. So with that, I think I will start with a little bit of data that we all know, right? From WHO.
Excuse me, Tom,
yes.
Me to interrupt, but you're showing this in speaker notes mode.
I'm sorry, my bad.
There you go. Good.
Yeah, so apologize. So I would first like to show the okay, so I was first like to show start with some of the WHO data. So as we all know, in 2000, right? I mean, compared to just a couple of years ago, 2019, there are, I mean, an increase. We understand that there's an increase of lifespan, right? So, I mean, like there are 6 years of increase, and at the same, actually, I have some technical difficulty. Yeah, I apologize. Okay. At the same time, right, we also understand that there's a the leading cause of death for there's an increase in mortality due to the age-related diseases. So now the question there becomes, in the first session, and we discussed that we all discussed that, there's, aging is a risk factor, right? And we also all agree that aging is malleable. And if we can treat aging, there's a good chance that we can prevent the onset of age-related diseases and eventually decrease mortality, right? And we also have a discussion from a clinical trial standpoint. We need a measurable point, right? We need an exit point. In order to run a trial. And currently there's, in a way, no good way in doing this, and this boils down to how we can measure aging, or how we can measure age. So to measure age, right, the most defined ones are those ones that have problems, right? Frailty, cognitive function, right? When the phenotypes or when the symptoms already appear. But it might be a little bit too late. There are also, we also had very good discussions about, different ways of measuring age. For example, can we actually develop different biomarkers, maybe wearable device for vitals, right? And also a great deal of discussions in various different topics that measure age. And I think one of the area that is very hard and upcoming is the AI machine learning approach, right? Many startups are working in this area and try to make use of this great, AI machine learning approach to measure aging. So now, okay, so now if we were to try to measure age, there are many different considerations that we need to think about, right? So for instance, if we were to do some kind of NGS work, we have to take a look at different tissues, and the extraction method might actually change the cell states. As soon as we take the cells out, there might be some dissociation, induce changes in the way how the cell behaves. And if we were to culture them, that's also another problem. The cells can change to a completely different states. So these cells can, we're culturing them in a medium, right? And these medium can actually, to some degree, reprogram the cells, right? So are they still the cells that we are thinking that they're when they were in vitro, in vivo? We may not be sure. And in particular, different lab has different culturing and extraction methods, right? Which makes it a little bit difficult to actually compare across. So we have also discussed many aging intervention strategies. So these are, there are even more variables adding to the equation, right? So in the session one, we have a great deal of discussion talking about partial reprogramming and a little bit about the safety concerns if we put it into practice, right? So and I think I would like to take this particular example to have a discussion, right? So for example, I just recently read this paper talking about some of the progeria mice. If you were to treat the mice with a single dose of Yamanaka factors, what happens is, when you treat them with just single dose when they are very young, and that will increase the lifespan and improve the body composition throughout life. So the question there becomes, when is the best time to conduct aging intervention? So I mean, of course, I don't have a good answer to it, right? I mean, when should we intervene? But I think, I mean, collectively, this will require many different researchers or research groups to look into it, and in order to answer this particular question. So now that presents one problem, right? So the problem is we have different we have a lot of aging-related publications, right? But there are actually a lot of different disparities in data collection in terms of, for example, some of the data that are in the paper. I mean, it is not entirely sure sometimes that, I mean, whether I mean the strings information is that spelled out well, right? I mean, the kind of diet, environment, age, gender, speed that they're I mean, the kind of information might be hidden in this kind of paper. So in my opinion, one of the big challenge is actually data integration and comparison. So taking back 20 years, right? That's when the time microarray was very popular, and at that time there was this standard called Miami, right? So this is the minimum information for a microarray experiment. So, and with that, I mean, it's basically collecting some of the description, you know, the species, the kind of information that will allow researchers to compare data across different microarray data across. So, you know, few years back or plus minus a decade back, so there are also NH-ENCODE project and NH roadmap project. So these are also added, you know, a bit more quality control, different kinds of normalization methods, you know, different standardization on protocols and data processing. So in my opinion, I mean, these are the kind of consideration for data collections if we were to want to improve data integration and comparison. So when I first talked to Taji about this talk, right? So we were asked to think about a future back approach. I mean, that I think data collection for aging research is one of those. And I mean, now, you know, look out in 10 years time, I mean, we ask the question, what do we want to analyze? And I think that what we need to do now is to understand what kind of information do we want to collect right now. So, and how do we collect? I think we need to have a better sense of what kind of minimal amount of information that we can collect. And so we have this metadata to basically enable our future. So with that, thank you for your attention.